Hello, I am Anders Lunde. I am the author of this paper here, which is called A Versatile Toolbox for Semi-Automatic Cell-by-Cell Object-Based Colocalization Analysis. And in this video I will take you through how to download and install this plugin for ImageJ and how to count cells in a semi-automatic fashion and how to count which fluorescent marker or other types of image signals different cells contain using the tools we have presented in this paper. So the first step is to download and install ImageJ. So you can go to find instructions here. You find this paper online. Just Google this title. And this is freely available online in this open access journal, Scientific Reports. Then you click download and installation and you get to this instruction here, and it recommends that you download FIJI, which is the same as ImageJ more or less. Let's go to this here, and then you click the version that is appropriate for your operating system. In my case, I am on Windows, so this will start to download FIJI on my Windows computer. Alright, now that FIJI has downloaded, you open the zip file and you can copy it to anywhere you like and extract the program FIJI demo. I make a folder and I put my FIJI download here and I extract all the files. This will install FIJI on my computer. Then I open FIJI, which is basically ImageJ with a lot of extra programs pre-installed, extra plugins I should say. The first time you open FIJI, it will probably look for updates of plugins, etc. No, apparently not. So the next step in the installation is to add update sites. So here it says click this sequence. Let's put this screen here. So, oops. Installation. So we're supposed to click help, update, and wait a little bit for look. And then you choose, here is the critical step, so you see then you have to go to manage update sites, click this, and then add an update site. So you click add update site and you add this link here as the URL. You see the URL here, and you can call this what is this? You need to provide upload information first. Okay, make sure you don't have the spaces in here. You have to remove the spaces in front, and then now it should work. And make sure that this is enabled, and you can call this colocalization object based, for example. I don't think this really matters. You don't have to even change this. And then you click close, and you see that these two plugins, which we will talk about in this video, are selected to be updated and installed. It says install it, so we click apply changes and it downloads the plugin and it automatically updates the plugin whenever there is a new update. So So that's the whole installation process. And it says please restart ImageJ. You click OK, and then you restart ImageJ. And now the two ImageJ plugins which we will talk about today have been installed under your plugin tab. Let's, for example, open the object counter program. There you go. Now I would advise you to make sure that this FIJI installation is always pinned to your Windows taskbar. So right click this and click pin to taskbar, and then this icon will stay there. Okay, now that we have installed FIJI and the plugins, we'll now go through the examples in this paper to see how we can use these softwares to analyze our microscopy images and to count the cells. So we will use some example images that are presented in the paper, and they can be downloaded from this link. So if you click here and you download this input image for example number 1 and 2, you can follow along with the examples used in the paper. So I have already downloaded this image, and I will now open it in ImageJ. It's a fairly large file. So this TIFF image is a set stack confocal scan containing 4 channels and 35 set slices, as you can see here. So these contain many cells that are stacked in a few layers, as you see, maybe 2 or 3 layers of cells on top of each other. And in the first channel here we have nuclear staining with the Huxt. In the second channel we have a fluorescent protein expression, TD tomato, in the subset of neurons. In the third channel we have a retrograde labeling of another subset of neurons, and as you can see, sometimes they overlap and sometimes they don't overlap, so we have different populations here. And we have also a fourth staining, which is immunostaining for a nuclear protein called LBX1, which is a transcription factor. And again, this will label some of the other neuron subsets and not label in some of the other subsets. So this is the question we have. This is the colocalization analysis we want to figure out. Which of these neurons are expressing these other markers, or in other words, more precisely, are they positively stained for these markers? And we will see now how these plugins that I have presented in this paper can be used for such an analysis. And especially emphasis here have been placed upon analyzing in such 3D data where you can have cells stacked on top of each other, because this makes it a little bit trickier, and we will see how we have tried to solve this problem. So we will see how all of this toolbox that is presented. Here is an overview of how it can be used. So we start with a multi-channel input image, which can typically be a set stack as well for 3D images, but you can also just handle 2D images. And the first ImageJ plugin, which I will present, is processing this input image into an output where it's easier for both humans and computers to recognize different colocalization situations. So this is an object-based colocalization analysis optimized output image. And with the second ImageJ plugin, we will quantify all these neurons and ask which fluorescent markers they are positive or negative for in a semi-automatic manner. And then we will also look at the Excel macro file, which can import such data from the analysis and can import from a large data set, so that you can have many sections throughout a brain or some other tissue. and combine that with a MATLAB script into a 3D visualization where the different colors represent different colocalization categories. In other words, different types of cells that express or are positive for different types of fluorescent markers or other types of signals, imageable signals. So a couple of things that this paper is trying to address is these two problems that you see here during analysis. And one of them is that when you have two cells that are stacked on top of each other but not actually colocalizing, then if you see them in a single 2D image, you will get a set projection artifact, a false positive. And this makes it difficult because of course it would be best to analyze the set stack and look at the whole 3D data, but this is very time consuming. And what this plugin is trying to do is to translate the 3D data into a 2D image, which it makes it easier to analyze, but without having these kind of artifacts. So what is actually happening is that the plugin chooses to only display either this green one or this purple one, so that it doesn't appear as they are colocalized in a 2D image. We will look at that later. Another problem which is typical for those that are analyzing signals coming from a subcompartment of a cell, such as for example transcription factors which are found in a nucleus, then you can have this problem here where the subcompartment is outside of the image data, and then it appears as a false negative in this case. And the plugin addresses this by for example showing only signals from the subcompartment. So all of this green would be ignored because this is not a reliable source to say that this cell is negative. So in this case we would be wise to only analyze the nucleus of every cell, and that is the only way we can say if a cell is positive or negative for a nuclear localized marker, is by having the nucleus being the proxy for the cell itself. So we should not look at the cytoplasm in this case. So we will see how we do that, how we can output only the nuclear signal and what's inside, and check out what's inside the nucleus with this plugin. So let's start up the plugin for which is called the first plugin, which is called Colocalization Image Creator, and we enable all these hotkeys, which makes it easier to use. And then you have this menu where we can transform this raw data into an image that is easier to analyze for us. And let's have a look at the paper which is explaining the first plugin. So in this figure it tries to explain how this plugin operates with the concept of image elements. So you can create your output, your processed output, as you see here and here, from the input image by adding what we call elements. So you extract some data from the original input and you transform that with your elements, which has different settings. If I click add element here, you see that we have two different types. We have binary element and grayscale element. The difference between these is that the binary element is a thresholded input channel. So you can only have a 0 or a 1 value of the output. As you see here, it's either completely black or completely full of signal. Or you can have grayscale elements, which can have many degrees of signal intensity. And for most analysis procedures, it's better to have binary signals, because then you can say that the signal is either present or not. But in some cases you might want to use grayscale signals, because it's not always easy to find the threshold that separates these two, and sometimes you want to look at the signal intensity itself. And it's also good for looking at the original data in your output. If you want to see how was it originally, you can use Grayscale for that. So if you go into either of these, then you will be presented with many options. You can set the color, you can select which channel you want to have as the input, and your method for thresholding this. It can be manual or it can be some automatic procedure, and you can turn on outlines on this. Let's look, for example, at channel 1, which was the Hux signal, and let's do a manual thresholding here. Let's present the output in white, and let's take outlines off for now, and we'll do a preview of this, and then we are asked to set the threshold, and let's say that we are happy with this threshold, and we click OK, and then we are presented with a preview which looks like this. And we can try to clean up this a little bit by adding some macro commands. For example, this sample here, we enable that and see how it looks, if it improves this, if it removes some of these dots here. I will set a new threshold value, so I remove this and I set a bit higher this time because I thought it was a bit including a bit too much, too many clumped cells together. And now let's see if these macro filters have removed all these dots. And the main thing I want to demonstrate here is that you can also convert to outline, and then you do this. Oh, I should have reused the threshold setting. And then all these signals will be converted into outlines, and later we will see how this can be very useful for visualizing what's going on inside of the cells. So that's basically how you create elements. Now I can click Finish, and this will be added to my output as a single element. So now my output image contains one element, and this is the input from channel 1. I can now add another. Let's say I add a grayscale element, and I add in green from channel 4, which is the antibody LBX1 staining, and I click Finish. I set my brightness value here. I click OK, and then now I have two elements in my output image, and let's see how these combine. Okay, they combine in this way. So now I have the outline of the nucleus for many of my cells, and I can see what's inside of the many of the nuclei. So some of them have the green signal, the LBX1 signal, and some don't. And this is one example of how you could analyze your image data by transforming to a bit more useful output. So what we see as the top slice here is actually a Z projection. But if you go into the, you can just go into the stack itself to see the original stack data. And here you can see in more detail what's going on. So for example, if the set stack is too crowded and you have some cells that appear merged, then you can go in here and you see that, okay, here is the first cell and here is the second cell. So these are indeed two cells that both seem to be positive. But as we can see, as we will see later, this sets a projection here, maximum intensity projection. You have some options to set for that. This is not the normal set projection. It's a special processed projection which tries to avoid this set projection artifact, and we will see later how that is done. Okay, so this figure here, this figure 3, tries to explain how the plugin number 1 puts together different elements, binary and grayscale elements, and outputs them to a channel. And you can have multiple output channels. I will show you that if we add another element. Let's say we add another binary element, and in this case we output it to a new output image channel, number 2, and we make it magenta, for example. And let's say we want to see what is the colocalization between the nuclear signal and the retrograde staining of the neurons. So we will, if we select more than one channel for the binary elements, then the output will be the Boolean AND between these two thresholded signals. This can be seen in this figure here. If you select two channels, here channel 1 and channel 2, and you intensity threshold both of these, then you will get the Boolean AND result of these. And then you can apply these macro filters. You can filter away large particles, convert to outline, etc., and end up with a signal that represents the overlap between these two channels. So I thought I could just show you this right now in this example by adding two channels and outputting it to a separate channel. Then I will, if I click finish now, I will be asked to set threshold. And by the way, you can also pre-process your images to binarize them beforehand with more advanced algorithms, and then you can use that as an input. And now you threshold both of these. So I was only asked to threshold one of the channels because the other channel, number 1 with the nuclear staining, was already thresholded from before, as you see here. You can change these thresholds, but you cannot use, at the moment in this plugin, you cannot use different thresholds for different elements. They all use the same. And here you see now that I have, in the output image, I have 2 output channels. So I can study this data with not only 2 image elements here. I can also combine this with this single image element in the second channel and go back and forth and compare and contrast these two types of processed data. So this was the colocalization between channel 1 and 3. And of course here you have all the nuclei in the white outlines, and you have I believe it was channel 4 in the green. So you already have a lot of representation of different types of colocalization events. And yeah, we will later see how we can use the second plugin to actually quantify and count these different types of colocalization events for the different cells. So this figure here is showing how you create an output image from creating elements, and that you have 2 types of elements, and that you can output to multiple output channels. And in this schematic here, it shows roughly the algorithm for how these elements are calculated and how the pixels are output to the final output image. So without going into details here, I will say that the first step is that all the grayscale elements are sequentially generated and added to the output image, and then all the binary elements are added to the output image. And that means that the binary elements will always be pasted on top of the grayscale image, because the idea is that the binary signals are more important in the colocalization analysis, because it says something about if the signal is there or not. We will get back to that. So this is showing more or less the algorithm that the plugin is using, and these are the 3 steps that the algorithm is doing when finding out how to show the grayscale element based on user input, and these are the same for the how the binary elements should be shown based on user input. So what's the threshold level? If there are multiple channels, it should be the Boolean AND between channels, and then apply filters, remove particles. You can also have a 3D filter based on size for advanced, if you go into the advanced menu and you can convert to the outline. Note that it's only the binary element that can be converted to outline. And you set the color and convert to RGB. That's the same also here. You convert to RGB, and then you add this to the image, that the output, which may or may not contain grayscale elements from before, and then all the grayscale and binary elements are added for a single output channel. So for example, there can be 4 grayscale elements and 5 binary elements in a single output channel, which should represent to you some information about colocalization events. And as you saw before, the different elements, here are 3 different elements. You set a single color for each of the elements, so you can look at the color to know what colocalization event this color and signal represent. So this is basically the algorithm. To talk a little bit more about the algorithm that determines how the final output image looks like, then you can refer to this figure here, which shows that there are a few simple rules that determines which signal should be output in cases where there are a conflict where a pixel is occupying the same location in the x, y, and z coordinate. So the first rule is that all binary elements are prioritized, so they are pasted on top by being added last. So you see here that even though there is a lot of grayscale signals, all of these binary elements, they are let's say getting priority over the grayscale signals. So be mindful of that. And then what if there are 2 or more binary elements in the same location? For example, here you see the white and the green signal is in the same location. Well, then the plugin consults the color priority table, which here says that white, or grays in other words, has the highest color priority. So white will be prioritized, and we will see how this can be useful. For example, if you know that all green cells, they have to be white also, like if in other words, if white is a superset and green is a subset, then there is no problem that you don't see this green signal, because you know, oh, it must also be green. Every white is also representing the green, the contents of the green. For example, that this white is represents nucleus and another signal, then and green represents only the other signal, but then you know that white of course has to contain the other signal because it represents both the nucleus and the other signal, whereas these are not nucleus. So that's how this can still be useful. And then you have you have the outline elements. So these are here competing. You see this is the same as this cell, and the only reason that this is not shown here is because red has a lower priority in this case. You can change this as you want, but otherwise the red would be on top here, and being outline element, you can see what's inside here. So that's basically the rules that determines what happens if a signal of the same element is in the same XYZ location. And you also have this 2 figures here showing how a grayscale image is output and what settings you have for the user. You can also add some macro filters here, and this figure for binary elements, what user options are available and how that will result in the output. So what I was just talking about was basically the element mixing rules, how different elements they mix together when they have the same position, and you can read more about that here. Now we looked mostly at 2D images and cases for 2D. Now what about the set projection output? Because as I said, that this plugin is one of the things that it is doing is to try to tackle this challenge here by providing 3D data as a 2D image, but where you avoid some false positives and some false negatives and some other cases. And just to explain that one more time, if we look at the output, so the set projection, this top slice, which is set number 1 of 36, that is a set projection. And if I go to the next one, then we look into this actual set stack. But the idea here is that let's try to look only at this top slice as much as we can, because then we don't have to go into the stack all the time. And of course this is means that this technique, this plugin, is more suited where you don't have a very large depth of cells, and you don't have very tight clusters of cells, because then your set projection would be full of cells and it would be very difficult to tell one cell apart from another. So this is more suitable for slices of tissue sections, and either extracted when cutting the tissue or extracted as substacks from a large 3D scan digitally. So let's look at how. So the main issue here is what to do when we see two cells stacked on top of each other. Let's see if we can find an example. These are fairly stacked on top of each other. So how should the output be? And then what we thought was the one way to do this was to say that, okay, we can't watch both of the cells at the same time in a good 2D representation, because that's simply not possible. So let's try to be as unbiased as possible and choose have some rule, the unbiased rule as to which cell to visualize in the set projection. And what you can do with this plugin is you can choose to output either the top or the bottom cell. So which one do you want to look at? So if you have a set stack here, as you see, this is an example output from the plugin number 1, and you have 4 cells stacked on top of each other. Then if you choose the topmost binary object, or just the top priority, then what you will see is only the top green cell. If you choose the bottommost object to be prioritized, then you will see only the bottommost prioritized object, be it outlined object or not. So you can study this figure and see some different cases and different rules, which are also described in the text. And but the general rule is that only the topmost or bottommost binary object will be displayed, and the grayscale output will never be prioritized if there is another binary object overlapping it, even if it's not topmost, because the grayscale signals, they don't have a clear definition of whether this is actually a signal or not. It's just to help you sometimes. And as we also explained, you should never use more than one color for a grayscale element. And you will get actually a warning if you try to do that, because then you will get this false magenta signal looking as a like a colocalization signal when it is in fact not. So you can study this and look at different cases what happens, but maybe one thing to note here, important thing, is that if you have an outline object like this outline, red outlines here, then if you see something in the set projection inside the outline object cell, you can be sure that this signal inside is coming from actually the same set position as the binary signal, as the cell in this case. So this will never be a false signal inside inside the binary outline, because we only show what is actually inside here. We extract these pixels and display them in the top here. So the only reason that you have here a false signal is because you have used two grayscale elements in the same output channel, and that is not recommended, and you will get a warning if you try to do that. So don't worry about that. Here you see, for example, if you use binary objects instead, then you avoid this problem. So of course the problem with this is that you get you will not see some of your data, because you will not see those cells that are stacked. You will only see the topmost. You will not see the bottom or the ones that are under. But at least you will get a unbiased selection under some conditions and terms, of course, if you consistently choose either the top or the bottommost object to be visualized. And we will see in the example how this can be useful. Now, these terms and conditions, they are mostly relating to that you should have that your different cell populations are uniformly distributed across the tissue, that there is not any bias towards the top or the bottom if you want to count and see the true proportion or ratio and number of these types of cells. And if they are very different in size and shape, that might also influence this technique. And yeah, and also if you are really unsure, then you should simply go into the set stack by scrolling the mouse wheel and look what is actually happening in the original data, in the original 3D data. But this might be a bit confusing, but it might be a bit clearer when we look at the examples. And by the way, this is another reason for using mostly nuclear signals, because mostly they have less irregular shapes and different shapes than many cells might have in the cytosol. So if you stick to the nuclear signals, which we will mostly do, then you get less variability in size and shape. Okay, so let's look at some examples of usage. So we will go through example 1 in this paper, visualizing colocalization between two neuronal cell populations with this colocalization image creator plugin. So in this example, we want to compare a population of neurons retrogradely labeled with a fluorescent tracer. So that is this channel here, and we want to compare this to a population of neurons expressing a fluorescent reporter protein, and that is this channel here. So we want to see what is the overlap between these cells and compare these two populations. So to do this, we will use two binary image elements, one to represent the colocalization of the retrograde label and the nuclei, and the other to represent colocalization of retrograde labeling and the reporter expression, this one, and nuclei. So as I explained, we don't have to use the nuclei, but because this helps with identifying individual cells easier, and since we will later look at the signals that are inside the nucleus, we recommend to do this. So let's do what this example is saying, and we will basically create two binary elements, and here are the settings for number 1, and here are the settings for number. So let's follow along with this example and create these two binary elements, starting with binary element number 1. So first we will delete all the elements that we had worked on up until now, and we will start with the first binary element. So here it shows that we should look at the overlap between all these 3 input channels. We want to show that in white. So basically, what is the colocalization between all these 3 signals? Where do we see a signal that appears in both the nucleus, and that also positive for this label and also positive for this label? We want to see that in white. And we want to make some filters to this output to do a minimum filter, and we want to do fill holes if there are some pixel holes in the objects, and we remove small objects that are smaller than 20 square micrometers. Click finish, set the threshold. It's generally a good idea to include a bit too much signal than too little, because this most of this will be filtered away by the minimum filter that we had, like so. Of course, this is dependent on the quality of your image, and you can do some pre-processing to do binarization, or even improve the signal yourself. So what we saw here was that this cell that is output in the original example was not really included. So then we can change the threshold. Maybe it's this cell here, so it seems to be okay for this channel number 3. Maybe the issue lies in channel that was a bit low, it appears. Maybe the issue lies in channel number 2. This has a weak signal here, perhaps. I put it a bit lower. So basically, this setting of the threshold is of course very critical, and you have to set a threshold yourself or do a binarization step that you believe is representative of your actual signal output, and you should always verify that this signal actually represents the true original staining. And I talk more about that in the section called verification channels a bit further up, that you should add this original data output and compare and see if you have a binary signal that represents the actual positive or negative staining. Of course, this is a problem in colocalization analysis overall. At least putting a threshold sets an objective value that you can consistently use, but it comes with some problems. That's why you should always use this verification procedure that you can read about. Cell as well. And of course we can verify this by adding some grayscale verification channels. I like to view them in white, so I want to see channel 1 output, which is the nuclear staining. I want to see how that actually looks in a separate channel, so I add that, and I add some brightness to it, and then you will see that I have created a new channel here. So this is the verification in grayscale, and now I can look at this cell. I go down, it is here. I go and see in the other channel, it's this nucleus. Yes, it is a true nucleus, but it is a bit weakly stained. But so my threshold is at least correct for this, because it should include this staining. So I go back to channel 1 in the set projection, and I proceed to add a second binary element here, which consists of channel 1 and 3 and should be viewed in green, and otherwise have the same types of filters applied. So the double slash is to disable or enable a certain command. And then I click finish, make sure it's output to this first channel. It reuses the thresholds from the first element because it's channel 1 and 3 is included. And then we get this final output in a minute or a few seconds here. So this was the verification channel, and here is the final output. So here we see the final output, and I have been adjusting a little bit the thresholds to better match reality, and this gives a very nice result. And as we show in the paper, well, first of all, let me say that I have also added the verification channels for all the 3 channels that we are looking at. These are of course grayscale, so they use these contrast values. So for example, if you want to see whether this is actually a signal that you should think is real, then you can go to the location of the original signal, and this is a green cell, so this should be positive for 2 of the signals, but negative for 1 of the signal, which the white is representing. So let's see. Yes, this is a nucleus, or maybe even 2 stacked together, but it has no signal in this channel, but it does have a signal in this channel. So it's very nice to use this original data and use the keyboard shortcuts to quickly jump between them to verify. And you can even do that in the set projection very quickly. You see that this does not appear to be a signal positive in this channel, even in the set projection. But here you see, yes, this cell here, this is indeed a retrogradely labeled cell. And what about this white cell here, for example? Let's have a look. Yes, it's a signal in all 3 channels. Very good. And if we go down to the set position to inspect a bit more carefully, yes, the nucleus is there. Yes, the genetic labeling is there, and the retrograde labeling is there. Even though the staining quality is quite low, you can still see that these 2 cells are positively stained in both channels, and the nucleus is part of this cell. And that's why you get a white signal here in the stack, and it's nicely viewed in the set projection. So if we go back to the article, here we compare what would happen if you do a traditional set projection with the 2 channels we are looking at, which was actually this cellular population and this cellular population. You would get a lot of false positive signals, like this white signals here, which are actually, these are not overlapping. If you go here, you see here and the other channel, these are not overlapping, but they appear to be co-localized because the other signal is on top of it, and that creates a false positive white signal here. So this is example number 10. You see that it is actually been removed here in this case, because the output from the plugin is correctly showing that this cell is only positive for this channel and not this channel, and because the other signal is coming from here top, but that is not the same Z position, so it's not part of the set projection output in this algorithm. And this is cell number 10, so here it's number 8, 9, 10. you see here in this orthogonal view that they are actually on top of each other. This is just a figure made for the paper. It's not actually an output from the plugin as of yet, but it's just to show that with this technique you can avoid set projection artifacts while identifying the true colocalizations here. So it's a bit difficult to understand maybe, but try it out with your own data set and see if you can make sense of it. So just to remember again that what you see in the top set projection here, if you use binary signals, you can be sure that these are not representing any overlap from stacked cells, because the binary signals are only coming from single locations here. So if you have stacked cells like this number 10, it will only show one of the signals, and this is just one example. I should have shown you also where you would have another signal here, for example blue or white or something, but then it would only show the topmost object, only the white or the blue or the green, never a mix of the colors, because binary elements, they never mix. So that was the first example, and you can see now how it's much easier to count these populations. You have 4 cells that are positive for all 3 of these markers, and you have a lot of other cells that are positive for only 2 of these markers, the nucleus and this population, but not this population. So of course this is not a complete analysis of all the different categories of cells, but this is just one example. And we also see that if you restrict your signal to the nucleus, you can have a better separation of the cells, because we use the Boolean AND between these thresholded channels, which means that you will only see the nucleus part, and that provides a better separation and easier identification of individual cells. And I recommend that if you see some weird shaped nuclei like this, and if you're extremely concerned about the accuracy of your total number of cells and not only the degree of what the colocalization categories exist, that you can go into the set stack and have a look, and also look in the verification channels and see what's actually going on, and you can use that when you count your cells in the second plugin. So let's look at the second example, visualizing colocalization of immunostaining, which is this channel 4 here, and retrograde labeling, which is channel 3, and reporter protein expression, which is channel 2, using this plugin. So this is slightly more complex, and we will use some of the more capabilities of the plugin. Most importantly, we will use this outline element. You see this is enabled here, and we will also use 2 different output channels and see how this can help us analyze this same picture. But now we want to also analyze this channel, so it's going to be slightly more complicated. Let me just check if I'm still recording. Yes. So we delete all of the elements that we are currently watching, and we simply we process this input image in this way that is recommended here. So we click add element and we follow these settings. So first we create binary element number 1 and 2. Let's start with number 1, and this is supposed to be in magenta, and we are going to watch these two, the overlap between these 2 channels, but we want to see the outline, and we apply the standard filters for our image. Now this is what we are visualizing here is the nuclear signal versus this retrograde neuron. So now we are essentially going to look inside of all the retrogradely labeled neurons, and we want to output it to channel 1. Click finish, We apply our thresholds. Remember that it should be a bit low for the nuclear signal. We learned that in the previous exercise. And this is the retrograde labeling. And we click OK. So now what we are seeing here are all the retrogradely labeled neurons, but we are seeing the nuclear location, and we're seeing what's inside of this. Currently we don't see anything else inside because we don't have any other elements, but we create also then what we want to see inside of here, and that is a grayscale element 3. Let's add element number 3. We'll do number 2 afterwards. So that here we should put, we want to see in white channel 4, which is the antibody staining, and we just make sure we can see all of the faint signals as well. And now we will have create this image here, which is all of the retrogradely labeled neurons, and we want to see which ones are positive for our antibody staining. And we see that these 4 cells appear to be positive. Coincidentally, the same 4 cells that were subpopulation of the overlap between the two different neuronal populations we looked at in the previous example. Let's continue to proceed with the example, and so now we have a very good way to look at this retrogradely labeled population, the magenta outline, and count which ones are positive for this signal, LBX1, but we also want to count which one are signal for which one are positive for the genetic marker. And instead of adding that information onto this image, which can be a bit confusing because then we have too many signals, we want to add it to a separate output image, so with that we have not so much information in a single channel. So let me show you what I mean. We will create again the same binary element from before, but let me enable the filters. But this time we will add it to a new output channel. So I will create a new one, and we will create this image here. We start of course with this binary. Let me see if everything is correct. Yes. So it will now create this new output. Yes, so now we have 2 output channels where we can see this retrograde population of neurons, and in one channel we see the LBX1 positive negative features, and in the other channel we don't see anything yet, but we want to have the information from the genetic tomato stained population, so we add as a grayscale element this information, and this time we don't add channel 4, we add channel 2, and we output it of course in the channel 2 output, and we just adjust the brightness for this grayscale and click OK, and we should be getting an image that looks like this. And now we can compare these 2 images. So this is the plugin 1, basically processing the input data into something that makes it easier for us to evaluate colocalization signals. So again, we see coincidentally that these 4 retrogradely labeled nuclei or neurons, they are positive for this signal from the genetic TD tomato expressing cells, and also for the antibody LBX1 staining. So there seems to be an overlap, and this didn't have to be like this. There could be some different here, but in this case it is like this. So this is an interesting finding, which indicates that the group of neurons that are expressing LBX1 transcription factor, they are also expressing this genetic marker, which in this case indicates a specific origin, a HOXB1 origin. So, and as I said, this example shows that it's good to output your images in several output channels, but otherwise that you have identical binary element here, so that you can quantify for each of these cells using the keyboard hotkeys Q and W, and you can see this cell is doubly positive, this one is doubly positive, this one is double negative, etc. And the good thing is that you don't have to rely so much on the 3D data. You don't have to go and look in the 3D data, because most of the time these signals that you see, this will be representative of the actual colocalization in the set dimension as well, as I showed you here, that because these are outline binary elements, they are outline, but yes, they are a binary outline elements. You will only, what you see inside of a binary outline element is guaranteed to come from a colocalization event in the X, Y, and Z. So as you see here, this is only showing the blue signal and not the red, so it's correctly not mixing these two signals, because there is a gap between these cells and it's identifying that these are separate cells, so we're only showing the topmost signal. And this is analyzed in detail in the paper in this XZ orthogonal view for all of these cells, and this is not part of the plugin output, it's just for the paper. But we see here that the first 4 cases here, these 4 cells, they are indeed true colocalization events, whereas in many cases we have avoided set projection artifacts, where the cell for this number 5, for example, which is this one here, this has some signal on top of each other in the HOXB1 channel, which is here, but this has been avoided. There is a slight bleed through here because of some slight inaccuracies here. These pixels here are carried over to the set projection, but it's rather clear, at least if you're familiar with your own dataset, that this is a negative signal, and so the set projection artifacts are indeed avoided. And there are several examples, many examples of these, where you would otherwise have set projection artifacts. Okay, so that was showing a lot, not everything, but a lot of what you can do with the first plugin, which is processing images into something more easier to deal with. I can also mention that you can, if you click Redraw Output, you will get some information about your settings. You can also save it and load it. You can change the color priority. You can change some set projection options. And you can also do batch processing, which you can apply certain settings and custom thresholds, etc., to a whole folder of images.